Bismillahirrahmanirrahim. I am Dr. Hafiz Muhammad Abdul Qayyum from University of Education Lahore, Faisalabad campus. The topic of my presentation is patterns of essential toxic metals distribution in the hair of lung cancer patients in comparison with healthy donors. First of all, introduction. Cancer. Cancer is not a single but group of diseases characterized by uncontrolled growth of cells and impaired cellular maturation. Cancer develops only in cells with damaged genes. It is a multifactorial and multi-mechanistic disease that represents a great variety of malignant tumor which can affect any part of the body. Cancer is the leading cause of death in economically developed and second leading cause of death in the developing countries. Almost 1 in 4 deaths were reported due to cancer in the world. In America, a total of 1.7 million new cancer cases and 0.5 million cancer deaths were reported in 2019. Whereas in Pakistan, an estimate of 0.5 million new cancer cases and 0.1 million cancer deaths were recorded in 2019. In worldwide, 14.1 million incidents of cancer and 8.2 million deaths due to cancer were estimated in 2019. According to WHO, by 2030, almost 22 million people will die from cancer all over the world. The stepwise development of cancer involves different stages including initiation, promotion, progression, and metastasis. Initiation is characterized by genomic changes within the cells, such as point mutation, gene deletions, and chromosomal rearrangement. Whereas promotion, the tumor development is promoted by survival and clonal expansion of these initial cells. In progression, consists of substantial growth in tumor size, whereas metastasis, cancer cells travels to other parts of the body where they begins to grow and form new tumors. Lung cancer. Cancer that forms in the tissues of the lung, usually in the cells lining air passages such as bronchioles or alveoli, and disrupts the proper function of the lung. It is number 1 killer in men and second in females. Overall, 1 in 17 deaths were reported due to this cancer. In worldwide, 3 million new cancer cases were estimated to arise. In Pakistan, around 20,000 new lung cancer cases diagnosed in every year. 5-year survival rate is only 14%. Types of lung cancer. According to WHO, lung cancer is classified as 2 major histological types. Number 1 is the non-small cell lung cancer and second one is the small cell lung cancer. Each type of lung cancer grows and spreads in different ways and is treated differently. Stages of lung cancer. Stage refer to determine how far the cancer has spread in local tissues and other sites of the body. These are used to develop a treatment plan and to define patient prognosis. According to World Health Organization, lung cancer is divided into 4 stages, stage 1, 2, 3 and 4, whereas stage 1 is the slow growing and stage 4 is the most advanced or highly aggressive. The risk factors which are involved to spread in lung cancer: tobacco smoke, crystalline silica, asbestos, radon gas, genetic factors, radiation, immune conditions, diet low in fruits and vegetables intake, and metal imbalance are the most important risk factors of the lung cancer. Significance of hair samples. Scalp hair resides in the dermal layer of the skin and is made up of 20 different cell types, each with distinct functions. The affinity of metals for hair is due to the presence of cysteine in the keratin structure as well as to follicular melanin, which are able to bind cations by ionic interactions. Concentration of metals in this matrix reflects from its mean level in human body during a period of 6 to 12 months as compared to other biological samples. Human hair has many other attractive disadvantages such as Final stability, easy collection, transportation, convenience store reflect long-term exposure to high metal and high metal concentration than in other bioindicators. Significance of selected metals. Essential metals are required for normal growth and development of human body, while their deficiency lead to various ailments. Therefore, metals play both curative and preventive role in combating disease. Trace metals are required at optimum concentration for proper function of the human biological systems. Calcium and magnesium are vital elements for skeletal formation and regulate heart rhythm and immune function, whereas cobalt is an integral component of vitamin B12, takes part in RBC function formation. Cadmium is a non-essential for human. It induces nephrotoxicity, immunotoxicity, and osteotoxicity. IARC classified cadmium as a group 1 human carcinogen, develops lung and prostate cancers. Chromium is an essential trace element required for maintenance of glucose and fat metabolism. Copper occurs in more than 300 enzymes. Its toxicity may lead to lung cancer, oral cancer, and ovarian cancer. Iron is mostly utilized in hemoglobin. Its toxicity typically damages cells in the heart. And manganese is necessary for brain and skeletal development. Sodium and potassium both work as a body electrolyte and control the transmission of nerve impulse and muscle concentration, contraction, and relaxation. Nickel is an important human urgent and develops inflammation, bronchitis, cardiovascular and kidney diseases. Lithium is very important in psychiatry because it acts notably at the level of neurotransmitters. Zinc is an antioxidant and anti-inflammatory agent. Strontium promotes strong osteoporosis, resistance bone, and reduce the pain of bone lesions. Lead toxicity may induce anemia, kidney damage, neurological effects, hypertension, and reproductive disorder. The association between occupational exposure to lead and lung cancer is well documented. Stress and cancer. Redox active metals undergo cyclic reaction, participating in the transfer of electrons between metals and substrates, and play an important role in the maintenance of redox homeostasis. Free radicals are known to react with all components of DNA, thus damaging its bases and deoxyribose backbone, causing mutation in crucial genes. Process of breakdown of metal ion homeostasis due to oxidative stress involved in a plethora of diseases such as cancer. Quadrant figure clearly demonstrated the mechanism behind the development of cancer. Major objectives of the present study: to examine the comparative distribution of selected metals in the hairs of cancer patient healthy subjects, to investigate the mutual relationships among the metals in terms of correlation coefficients, to evaluate the comparative apportionment of essential toxic metals, to investigate the viable variations in the metal levels with food, with habitat, food, and smoking habits in the hairs of cancer patient healthy subjects, to determine the significant differences in the metal concentration in types and stages of lung cancer. Experimental methodology consists of following 4 steps: sample collection, sample preparation, metal analysis, and the last one is the statistical analysis. Collection. The hair samples were collected on volunteer basis from the newly diagnosed cancer patient admitted in Punjab Institute of Nuclear Medicine, Peenam, Faisalabad. Samples from the healthy subjects were also collected. Information regarding donors' diet, age, abode, smoking habits, health status, medication, occupation, cancer type, stage, etc. was recorded on a pro forma. Sample preparation. First of all, washing of hair samples. The samples were cut into small pieces of 2 to 3 cm length and mixed with 50 ml of detergent solution in a conical flask. The samples were washed with plentiful tap water. It was followed by addition of 30 ml of triton standard solution and again shaken on an auto shaker for 30 minutes. It was finally rinsed with double distilled water. So the acid digestion of hair samples. The digestion was carried out on hot plate. An accurately weighed portion of the hair sample was treated with 10 ml of concentrated nitric acid and heated at 75 centigrade for 40 minutes. It was cooled to room temperature, followed by addition of 5 ml of perchloric acid with subsequent heating to soft boil until white dense fumes evolved, marking the completion of the digestion process. Metal analysis by flame AAS. Following selected metals were analyzed by flame atomic absorption spectrophotometer, implying optimum analytical conditions. This table illustrates the optimum analytical conditions maintained on flame atomic absorption spectrophotometer. Limits of detection and quantification of metal along with certified and measured concentration were also noted. Statistical treatment of data, minimum, maximum, mean, median, standard deviation, standard error, skewness, kurtosis, correlation, and quartile distribution were also noted. Results and discussion. Basic statistical distribution parameter for the selected metal concentration in the hair of lung cancer patients are shown in this table. Most of the metal exhibited a large range of concentration as given by minimum and maximum levels. On the average basis, highest concentration was observed for sodium, followed by potassium and iron, while lowest concentration was observed for manganese as well as cadmium. A random distribution pattern was exhibited by most of the metal as demonstrated by high standard deviation and standard values on one side and distinctively dissimilar mean and median levels on the other side. Basic statistical distribution parameters for the selected elemental concentration in the hair of lung cancer patients are shown in this table. On the mean basis, highest concentration was observed for sodium, followed by potassium and iron, while lowest concentration was noted for manganese and cadmium. Maximum asymmetrical distribution pattern was observed for cobalt and lithium, as supported by large skewness and kurtosis values. Correlation study of selected metals in the hair of lung cancer patients are shown in this table. Metal data were also subjected to correlation study in order to find out the mutual relationships and are shown in this table. In the hair of cancer patients, bold values indicate a significant correlation among the selected metals such as iron, potassium, chromium, cadmium, chromium, lead, which indicated communal variation in the hair of the patients. Correlation study of selected metals in the hair of healthy subjects are shown in this table. Similarly, bold values indicated significant correlation were observed among the selected metals such as potassium and iron, manganese and nickel, and chromium and cadmium. Red bold values represented the negative significant correlation among the selected elements such as lithium and sodium and Fe and sodium. Another important dimension of the present investigation is the multivariate analysis. Quantitative information regarding multiple relationship of these selected metals were assessed by principal component loadings. Principal component loadings of selected metals in the scalp hair of lung cancer patients is shown in this table. PCA, principal component analysis, yielded 5 PCs of the metal with cumulative variance exhibiting 89% of the whole data. PC1 yielded highest loading for magnesium, potassium and iron. PC2 yielded highest loading for copper and nickel. PC3 yielded highest loading for calcium, cobalt and cadmium. PC4 yielded highest loading for sodium and zinc. PC1 and PC2 were regulated by internal body metabolism and also controlled by environmental pollutants, particularly transportation related factors. Principal component loading of selected metals in the hair of healthy subjects is also shown in this table. In the hair of healthy subjects, PCA yielded 5 PCs of metals with cumulatively exhibiting 84% of the total data. PC1 yielded the highest loading for sodium, potassium, lithium, chromium and cadmium. PC2 yielded the highest loading for zinc and iron. PC3 yielded the highest loading for calcium. PC4 yielded the highest loading for magnesium and cobalt. PC5 yielded the highest loading for copper and nickel. It was also found that the loading pattern of selected metals was appreciably different as noticed in case of patients. Another important aspect of the present investigation is the comparative evaluation of selected metals based on demographic characteristics. Habitat-based variation of selected metals in the hair of lung cancer patient and healthy subjects as shown in this bar graph. The mean concentration of potassium, copper and lithium were found to be markedly higher and mean concentration of strontium chromium was lower in the hair of lung cancer patients as compared to healthy subjects. Comparative average concentration of selected metals in the scalp hair of lung cancer patients and healthy subjects based on food habitat is shown in this figure, which revealed that average concentration of calcium, potassium, iron, copper, nickel and cadmium were significantly higher in the hair of vegetarian and non-vegetarian patients than vegetarian and non-vegetarian healthy subjects. Difference based on smoking habits. In the scalp hair, average concentration of potassium, cobalt and cadmium was significantly higher in smoking patients than smoking healthy donors. Salient findings. Most of the selected metals were randomly distributed, but randomness was comparatively higher in lung cancer patients. Mean levels of copper, cadmium, lead were significantly higher in the hairs of lung cancer patients. PCA and CA revealed dissimilar apportionment of selected metals in the cancer patients compared with the healthy subjects. Here is the last slide. Recommendations. Body burden of other essential toxic metals should be explored. More clinical studies should be conducted, and future research should be focused on the relevance of respective mechanism. Acknowledgements. First of all, thanks to the organizing committee of this very special conference. I am grateful to the Department of Chemistry, University of Education. I am also thankful to the Head of Department, Professor Dr. Muhammad Abdul Rashid. Thanks to my lab students and staff members, and very special thanks to the staff of Peenam Hospital, Faisalabad.